All right. Welcome everyone to our first journal club of this new series, which is a Foundations of Neuroengineering series. Today we have Dr. Reza Shadmehr, who is a professor in biomedical engineering here at Johns Hopkins. We'll be presenting about the cerebellum and kind of a machine learning kind of perspective on the cerebellum. We're very excited to have him. He is a great teacher and kind of explainer of science, and so I think he's gonna we're really excited to have him, and I think he'll do a great job. So a couple logistical things before we start. I'm recording the talk, as you guys see, so just letting you guys know, and if you don't want your image shown, just keep your video off. When the main lecture portion is over, I'll end the recording, and then when we get to the discussion, if you want to turn your cameras back on, then you're welcome to do so. Another thing is that sometime in the middle of the talk, we're going to be sharing a poll about the event. So please fill that out when you get a chance. Helps us collect good information and understand how people want to get involved with TNT. And for those of you guys who haven't been to a TNT event before, TNT is the Translational Neuroengineering Technologies Network. So from the name, we're a group at Hopkins interested in translational neuroengineering, and we kind of explore these things through talks like this, journal clubs, seminars, and then also social events, and just kind of getting opportunities for people interested in neuroengineering to get a chance to meet and kind of further strengthen the neuroengineering community at Hopkins. So that's a little intro if you want to get involved. But I think that's enough introduction. I think we're ready now for Reza to take us on his journey.
Thank you, Mark. It's good to be with you guys. So I'm Reza Shadmehr. Thanks for taking the time to coming today. The pictures you see on the screen are some of the my joys in life. The top picture, this is a picture we took, I think, just on Tuesday, so yesterday. This is it was a little birthday celebration for two of my students, Paul Hage and Jay Pai, two PhD students. Behind him is Simon Orozco, also a PhD student, all of them in BME. Amin Fakharian, also a BME student, first year. Alden Shoup, who is going to be a BME PhD student coming in. And so these guys are responsible for almost all the stuff that I'm going to show you, in addition to Inkyu Zhang, who's a master's student in the lab. And the guy down here is Ehsan Sadaqatnejad, who just finished his BME PhD a few months ago. And he's really the person that put together the lab that made the work that I'm going to show you possible. So it's a real privilege to be at Hopkins, because honestly, I think that we get such fabulous students, and really the science is only made possible because of the dedication and the quality of the students that we're privileged to mentor. So I'm going to talk about the cerebellum. But before I talk about the cerebellum, I'm going to tell you about a very simple idea. And that idea is this. So in biological systems, probably in a way that is opposite of what you would find in artificial systems, learning is easy for biological systems. What's hard is unlearning. Maybe the opposite is true for artificial neural networks. So let me show you an example of this. So this is a honeybee, and they're really beautiful animals. Obviously, we depend so much on them for pollinating plants, but they're also a simple animal to use a behavior to test the idea and memory. So what you can do is that you can train the honeybee to associate an odor, let's say carnation oil, with nectar. So you give it the odor, and then you present it with the nectar. And so what he does is that he responds by extending his proboscis and getting that nectar. Now it makes it so that if you do it once, if you do it twice, if you do it 3 times, what happens is that on the third time, the third session of training, the animal will predict that, whoa, this carnation oil is really great. I'm going to get nectar for it. So it's going to extend its proboscis upon presentation of the carnation. This is just a form of classical conditioning. All right. So this is how we do learning. And then what you can do is that you can, for some reason, let's see what happened here. You can look at the retention of this memory that was acquired, and you see that, an hour later, 2 days later, the animal remembers. Now you can take another group of subjects and train them on the same task, but then do what's called extinction training. So we're going to try to get them to unlearn that this odor really is not going to be associated with nectar. So what you do is that, you present the odor, but you don't present the nectar. And this is called extinction training. And so what you see is that session after session after session, the animal learns not to extend its proboscis. Now what you do is that you come back an hour later after you did the extension training and you provide the nectar, and something really remarkable happens. You see that it's a little bit higher than what you left off, the memory. And then if you wait another hour, it gets a little bit stronger. And it's really remarkable that with the passage of time, that extinction training that you did seems to almost vanish. It's as if it didn't happen. And what this is called is spontaneous recovery of memory. So basically what you learn is that extinction training didn't erase this previously acquired memory. Somehow it masked it. And with passage of time, that initial memory that was acquired, it just comes back again. It's called spontaneous recovery. Okay. Now, this is really a ubiquitous concept in memory research. And I want to show you in humans now, in a very simple task that one can study in a laboratory, you get the same effect. So let's look at this concept in humans. We're going to do it in motor adaptation. So what happens is that you ask a person to look at this dot here, and then you provide a target. And then what they do is that they move their eyes from here to here. And that's called a saccade, like this. You move your eyes quickly from one side to the other, and your eye now ends here on the target. So you just did a targeted eye movements called a saccade. But what you do is that when that saccade takes place, instead of providing the target here, as the eye is moving, you jump the target and you move it up here. So the eye ends here, but the target is here. So that means that there's this error, okay? And you do this repeatedly. And what happens is that the brain learns that if you give me a target here, instead of moving my eyes to this, I'm going to add a little bit of commands to my eye so that it just veers off a little bit up. So that'll be the beginning of my learning. And then if you keep on giving me those errors, I'm just going to make these curved trajectories with my eyes. And these curved trajectories are indeed what happens. So if you look at the behavior, it looks something like this. So here's the perturbation. At the beginning, you don't have any perturbation. So the eye just moves to the target. There is no vertical component to it. But now you jump the target upward. So you get this error. And so the brain slowly learns to alter the trajectory of the eye. And if you keep up that error, what happens is that the eye keeps curving more and more with the training. So you've taught the brain that if you give it the stimulus here, move your eye up to there. All right, fine. Now let's try to erase this training that we just did. So now instead of jumping it upward, we're going to jump it downward. So at this stage, you just learn to move like this. But now when I give you this target, I'm going to move it down like this. So now your error is in the opposite direction. So we reverse the error. All right, what happens? Well, you unlearn. You move your eyes, unlearn that additional thing that you did. And what you see is that, well, here was a perturbation. Here's your eye comes back down more or less to where it was when it started from. Okay, now what we're going to do is that we're going to remove these errors. We're just going to give you this target here. And as soon as you move, we're going to erase that target altogether. So you won't have any errors to learn from. And what happens is that there's this remarkable thing, which is called that spontaneous recovery. Trial after trial, the eye curves back toward that initial adaptation that it had. Look at this. So it just recovers part of what it learned before. So you see this spontaneous recovery memory, even in humans, in the simple adaptation task. So as I mentioned to you, this concept of spontaneous recovery, the idea that unlearning is hard, it's true in lots of things. So for example, memory of fearful events. So if something happens that causes this anxious moment in your life, you fear it, and then, for the next 15 times, it doesn't give you that anxiety. Nevertheless, with passage of time, one can still have this recovery of that memory of the fearful event. people have done these kinds of experiments in motion adaptation. They've done it with respect to other kinds of adaptation, like adapting to prisms, eye movements, like I've been showing you, visual contrast adaptation, movement of your arm, reaching, and so forth. And in all of these cases, you see these concepts of spontaneous recovery, like what I showed you in the honeybee. So you can ask a simple question: why does memory exhibit this really, I think, fundamental property that there is, you try to unlearn, but it doesn't really seem to erase that memory. What it does is that it masks it for a while, and then after a while, the original memory seems to just, come back. So what's going on? How do we understand this? So let me stop for a second and let's see if you understand the basic idea so far.
I do have a basic question.
Yeah.
What when you present these different tasks, what's to distinguish from when you're learning versus unlearning, or like why do you learn in the beginning? Like what's unique about those tasks that they're learning? Like there's a whole history that the subject has before the experiment. How do you know that they're learning something novel to begin with?
Yeah, that's a really good point. So in fact, learning is never anything, really, you're right. So how do we know learning isn't really unlearning something in the past? You're absolutely right. All I've really shown you is the recent history as having errors in one direction and then errors reverse and go in the opposite direction. That's really all I've shown. And the idea that when the errors reverse direction, it doesn't really seem to erase what you learned in the past.
I have a quick question. So when you reverse the error, how
Yeah.
long are you trained on the reversed error?
Typically, it takes less time to train on the reversed error. So in these adaptation experiments, learning is slow, but unlearning seems to be pretty fast. In terms of bringing your performance back to baseline.
Okay. But if you train for the same amount of time as you train the first error,
Yeah.
still have spontaneous recovery?
Yeah, it's a good question. So what happens is that if you train as much as you did initially, then you basically become you basically become this learner of this reversed error, whatever that is. So in the case of if you go back to the honeybee, if you look at the honeybee, actually they trained it even more. You see that? So the extinction was actually even longer than the initial training.
Gotcha. Thank you.
No, sure, of course. Good questions.
Okay. One question.
Yeah.
So does it depend how much time has elapsed since the extinction training?
Yeah. So definitely, these are all complexities of that basic idea. Yeah. So and it depends on how much extinction training you did, how long you waited. So both time and the kind of the duration, how much time has passed, and the unlearning, the sort of extinction training that you were doing, that those are important parameters. So let me show you a simple model of this idea. And the model is like this. It says that any kind of learning that you do is going to be a cooperation between multiple systems. And it goes something like this. So you have your brain it's going to make some kind of prediction. And then there's this actual consequence of that prediction that takes place. So this environment provides you with the sensory feedback. You made a prediction. The difference between those two is your error, and you're going to learn. You're going to acquire a model of this environment. Now, the system that is learning is made up of many things, and some of those things, neurons, regions in the brain, however you want to think about it, some of those units learn a lot from this error, whereas there's going to be some other units that can only learn a little bit from this error. And so the idea is that there's this cooperation that's taking place between multiple systems meaning either regions in the brain or even neurons within the same region of the brain. For whatever reason, there would be different sensitivities to this error. So let's imagine a very simple scenario. So suppose we have two systems. I'm going to call one of them a fast system, and I'm going to call the other one a slow system. Both of these two systems are going to make predictions, and my prediction about what's about to happen is the sum of these two systems. And the superscript n is just a counter that says what trial I'm in, trial n. So this y hat f is coming from what I'm going to call a fast learning system, and this y hat s is going to come from what I'm going to call a slow learning system. Okay, that's the basic idea. Suppose you have a fast learning system and a slow learning system. Now, when you make a prediction, you compare it. Here's your prediction. You compare it to what the world actually gives you. The difference between those two is your error. Now, what we're going to do is that we're going to do something special about this error. This error is going to teach both the slow system and the fast system. Here's the fast system. It's going to learn from this error here, and here's how much it's going to learn. But then it's going to also forget a little bit of the learning that it's done. Similarly, the slow system is going to learn from error, and here's how much it's going to learn. But it's also going to It's also going to forget a little bit of what it has learned. And here's the key idea. Imagine that the fast system learns a lot from the error, but also forgets. On the other hand, suppose the slow system learns a little bit from the error, so it's a slow learner, but what it learns, it retains well. So that's the basic idea. All right, so now let's just simulate this very simple system. So we have a fast system that learns a lot from the error, but it has forgetting problems. We have a slow system that doesn't learn a lot from the error. It learns just a little bit, but what it has is the advantage of retaining what it has learned. So this is how you typically would learn. You would learn something, and this would be the output of your system, the sum of the fast and slow. And as it's learning from the error, you have this fast system that learns quickly, but you see, can retain, and then you have a slow system that learns slowly, but it has good retention. So the sum of these two is what is this output, the adaptation. Now, what's extinction? Extinction is reversing the error. So you make the errors in the opposite direction, and you make the system come back down to baseline. This is the output of the system, comes back down to baseline. But really what that does is put up a competition between this fast and slow system. So because the fast system can learn a lot, it goes negative. Because the slow system can't learn a lot, it goes a little bit down. The sum of these two gives you this red. But you notice at this state, I'm not the same state as here. So even though my behavior is the same, the slow state is up here, the fast state is here. Now, what's spontaneous recovery? Well, just remember that, my battery is running low. I apologize. Okay, fixed. Okay, so what's spontaneous recovery? Well, spontaneous recovery is the following. We allow time to pass. Now remember, the fast system has trouble remembering, so it rapidly decays. The slow system retains well, so it slowly decays. The sum of the slow and fast, lo and behold, is that spontaneous recovery that we saw. So mathematically, we can write down just incredibly simple equations that describe learning as a cooperation between two systems, fast and slow. And that the two are going to learn, and if you try to reverse the error, the two are also going to try to unlearn. But what happens is that unlearning is going to cause more input on the fast system than the slow, but passage of time is going to make the system go back to the slow system, which was the one that was principally responsible for what you did during adaptation. That's the basic idea. Do you have any questions so far? All right. Now,
Well, I do have a quick question. So
please.
that means the slow system, since
Yeah.
it will also like decay a long time. So the spontaneous recovery will also decay a long time. Is that?
Yeah. So spontaneous recovery would have a time scale that looks like this. It rises up, but it's not going to stay up forever. It's going to slowly fall down.
Okay.
Yeah.
So this is what we see.
Well,
previous experience.
actually, it's a little bit more complicated, right? I mean, it's not, so this is just with two states, right? So in reality, you may have more than two states, and the time scales you can vary. But the basic concept I want you to understand is that if you had different sensitivities to error and different retention properties in these two imaginary systems, then in principle, you'd have a way to think about why unlearning is difficult. All right. Now, this is just math, right? We're not doing anything about the brain. We're just imagining in principle how it can happen. So I haven't told you anything about the cerebellum, right? I've been just talking about this interesting phenomenon of spontaneous recovery. It's because we discovered these things, as you can see. It's almost, it's been 15 years. So this work was Maurice Smith, my second graduate student that did this work. And I mean, he's now a tenured professor at Harvard. So he's in his late 40s. So Reza, what have you been doing since then? The idea is that the math inspired us to ask, well, what part of the brain is doing this stuff? And can we have a task that we can study that would engage that part of the brain? And we can ask, well, how are the neurons actually, what are the neurons doing? And what's the spontaneous recovery that you're talking about? And do we see it in the neurons? What's going on? So the next thing I want to show you is that we've been studying a particular task, this saccades, because they show spontaneous recovery. And we know it has something to do with the cerebellum. So let's go back to our saccade. So remember, the saccade was, you look at this dot here, I present you with this stimulus here, you make a saccade, you move your eye from here to here. And then I'm going to give you this error. And so what happens is that, if you're a healthy brain, you're going to improve your movements by curving your eyes to move upward. So this is your adaptation curve. And then we're going to, reverse it, the error. So we're going to cause extinction. And then we're going to remove those errors. And then you see that there's spontaneous recovery. So, okay, we have a paradigm that we can see spontaneous recovery in healthy people. Now, if we now look at a patient population, these are people with cerebellar damage. What happens is that, well, they just don't show much adaptation, that they don't need to de-adapt more. Of course, they don't show much spontaneous recovery. So adaptation, learning from error in this task seems to depend on the cerebellum. So look, this is 14 years ago. Menanju Wilson was a graduate student, another BME PhD student in my lab. She said, well, look, this stuff depends on the cerebellum. We've now seen that you show spontaneous recovery and the learning of it depends on the cerebellum. All right, great. Okay, so then we thought, all right, so we need to understand what is the cerebellum doing to control the eye movements, to cause this learning and so forth. But the problem is, you can't really just study learning without understanding control of movements, because when you look at people that have damage to the cerebellum, it isn't just that they have trouble learning, they have trouble making movements. And on top of that, they have trouble learning. So let me give you an example. So here's a patient that has damage to the cerebellum. I want to show you his eye movement. He's going to make saccades from left to right and back. Okay, so what do you see? You see that he's moving his eyes from left to right and back, but he's not able to stop the movement of his eyes at the right time. So his eyes are moving, but they're overshooting, and then he's correcting, and then he's correcting, and then he's correcting, and he finally is able to stabilize. He's making a saccade, but he's overshooting. So he's correcting, and he's correcting. So it is this maddening difficulty that the brain has in controlling the eyes in order to make a stop on the target if there is damage to the cerebellum. So we wanted to study learning, right? We wanted to understand that spontaneous recovery. We thought it has something to do with the cerebellum, but when you look at what the cerebellum is doing, it isn't just learning. It is making predictions that are important for just control of the movement, which then manifests itself also in problems in learning. Okay, so we needed to step back and say, what is the cerebellum doing in controlling these eye movements? If it's not only learning, it's doing other things as well. So I need to now step back and show you something about control of movements by the cerebellum. The important thing to realize is that in order for you to move your eyes, you have this evolutionary long history of circuits that allow you to make movements. So there are direct projections from your retina to an area in your brains, in the midbrain called the superior colliculus. So this is not going to the thalamus and the visual cortex. This is directly from your retina to the superior colliculus that has the machinery in the brainstem called burst generators and the motor neurons on the eye to generate those saccades. Now, there is this supervision of this circuitry by the cortex. So left to its own, you would be like a frog that any stimuli that would appear that would attract your attention from your colliculus, you could move your eyes and your head toward it. But you're not allowed to do that because you have a cortex that evaluates the stimuli around you and decides what's important for me, what's valuable for me to look at. And through that value function, it determines whether the colliculus will be allowed to make a saccade to one direction or another. All right, so what does this have to do with the cerebellum? Well, the cerebellum is like the side loop. So this circuitry is a control system that can generate movements. Its commands are sent to the cerebellum. The cerebellum is listening to these commands and is making predictions. And those predictions are fed back that seem to be really important for telling the brain, telling the rest of the brain, telling the brainstem, when should I stop the movement? When should I, how should I produce the correct motor commands to end this movement properly? And so, of course, when there are errors, those predictions change, and so does the behavior. Now, one of the founders of biomedical engineering at Johns Hopkins was David Robinson. And David Robinson described these circuitries. He did experiments with it. And you know, there are many giants in the field of neuroscience that have since David Robinson described this circuitry. Similarly, there are people there are giants in the field of neuroscience that have described these circuitry. And similarly, there are giants that have described this circuitry, the cerebellum. So you might ask yourself, after all these years, Reza, what in the world is left unknown for you to come and tell us, to talk to us about? Well, I want to show you that there is still a really fundamental problem in understanding what the circuitry is doing. The basic problem is when you measure the neural activity in the cerebellum during movements like a saccade, it's really hard to figure out what is it predicting. So, you know, the cerebellum seems to be important for control of movements. It seems to be important for learning from error. But as I'm going to show you, it's not at all obvious what the heck is going on in terms of making the predictions. So although in patients, we see very clearly deficits in movements, we see clearly deficits in motor, in learning, when you look at the neurons, as I will show you, it's pretty darn hard to figure out how does this behavior connect to this neuron. So I realized that to understand learning, to understand that problem of spontaneous recovery, all that stuff, I first needed to understand what the heck is going on in the cerebellum. So what are these neurons doing to make these predictions? And so that's where I want to take you. Okay, so what's nice is that compared to the cerebral cortex, the cerebellum is easy in that it's basically a three-layer network. You have an input that comes to it, those copy of those commands that are being sent in the brainstem is sent to the cerebellum via something called the mossy fibers. That becomes the first layer, goes to something called granule cells, and then it goes to the second layer, the hidden layer, Purkinje cells, and then it goes to the output layer, the nucleus cells. So that's basically a simple three-layer network. That's kind of nice. You know, we can deal with three-layer feed-forward networks pretty easily since the work of Hinton in 1980s. So that makes this prediction, and then that prediction is sent as an inhibition to the inferior olive. It's a region in the brainstem. Inferior olive also receives what we think is excitatory inputs regarding the observations. The difference between what you predicted and what you observed becomes your error, right? That's prediction error that gets sent back. Now, what's really special about the cerebellum is that these Purkinje cells, which are receiving two kinds of inputs, they're getting inputs from the granule cells and they're getting this input from the inferior olive. It's one of the very few places in the nervous system where you can dissociate these two inputs. And it goes as follows. I'm going to show you that the prediction error can be easily dissociated from the other inputs that the Purkinje cell is receiving. So here's a recording in one of our marmosets, and I'm going to show you spiking activity in a Purkinje cell. I'm going to show you what the prediction error looks like as compared to other things. So it's going to produce spikes. This Purkinje cell is going to produce spikes. Here are the spikes that it's producing. And let's watch as these spikes are generated. Okay, so you see there are two kinds of spikes. There's this big old fat spike here. It's called a complex spike. That's this prediction error that's being sent to the Purkinje cell. And then there are these other spikes, these simple spikes. These are the outputs that are from the Purkinje cell, which are driven from the inputs that it got from mossy fibers and the granule cells. So, okay, we can see clearly the prediction information, what error information that's going back to the Purkinje cells. That's kind of special because in this neuron, we can see the prediction errors that are conveyed and that produce what's called complex spikes, these broad dendritic spikes that are generated in the dendrites and the soma of the Purkinje cell that are easily dissociable from other kinds of outputs that the Purkinje cell is producing. So we can measure the error signal that's being sent to the Purkinje cell. Okay, so then what's the problem, you might ask? Because, you know, now that you have this stuff, what's the issue that you're having? What I'm going to show you is that it's really hard to understand the output of these Purkinje cells. So here's one of our animals making saccades. So what you see here is the eye movement of this animal. So here's the saccade to the left. This is the velocity of the eye. Over here, this is the velocity. This brown thing is the velocity of the eye. So here's when it begins, here's when it ends. Here's saccade to the left, here's saccade to the right. Here's the output of one Purkinje cell. And you see those simple spikes that I was showing you. And so this cell is firing at about 50 hertz when the animal is just fixating, not moving. And then when the animal makes a saccade, this cell is pausing and then it releases the pause. But you notice the pause is lasting a heck of a lot longer than the saccade itself. Here's another cell. It's also pauses. This one does a shorter pause. This one does a pause and then a burst in this direction. This one does a little bit of a burst in that direction. This one does a burst, then a pause like this. And then finally, there's a couple more cells. So you see there's just this gigantic diversity of activity of these Purkinje cells associated with this movement. And that's been the puzzle. So how do we make sense of this relationship between the output of these Purkinje cells and the movement that's taking place? Okay, so what's the main idea? How do we put this story together so we can understand coding in the cerebellum? It goes back to the concept of error, those complex spikes that I showed you. The idea is we think that Purkinje cells, we and many others, think that Purkinje cells form groups that project onto a single nucleus neuron. So there's about 50 Purkinje cells that project onto a single nucleus neuron. Well, what determines which 50 these cells are? The theory that we've proposed and we've been using is that the cells that project onto a nucleus neuron have something in common. They all receive similar error information from the olive. So they all care about a particular kind of error. So that's the basic idea, that P cells that belong to a population are identified because of the kind of error that they get from the inferior olive. So they care about errors, say, to the left or upwards or rightwards or so forth, but that they form these clusters that are identified by the error that they receive, in this case, from the inferior olive. So let me show you more about this idea. So what we're going to do is describe to you what is this concept of this error and what's the biological nature of it, and then we're going to organize the cells based on this preference for error. So let's step back a little bit and ask, what do you mean by an error? So suppose you're this monkey and you're looking at this light here, and this is the map of the superior collector. This is how the visual information on your retina maps on to the space of the superior colliculus. So when I'm looking at this target here, what happens is that for me to hold my eye still, there's activity in this region of the superior colliculus called the rostral region. So there's neurons that are active here. Now, when you show me a target here, that's mapped onto this part of my superior colliculus, is to the right of my fovea, which falls on the left of the colliculus. So I make a saccade, and for me to make a saccade, activity goes up here, and then when it ends, my eyes are here at fixation again. So I'm expecting at the end of my saccade that the activity should be here on the superior colliculus. The stimulus should be on my fovea, but actually it's up here. That means that it's activating this region of the superior colliculus. So I'm describing the visual map of space as if there were light on the fovea, which causes activity on specific parts of the colliculus. So if I'm expecting at the end of my saccade that the activity, that basically the stimulus should be on the fovea, which means the activity should be here, it's actually here. The difference between those two is what's called an error. So the activity is not where it should be. So error produces unexpected activity among specific parts of neuron. These neurons in the specific part of the superior colliculus. Now, the superior colliculus maps onto the inferior olive, which then produces those complex spikes that I was showing you in the cerebellum. So what I'm saying is that there are some Purkinje cells that care about errors here, whereas there's some other group of Purkinje cells that care about errors someplace else. Now, what we're going to do is organize the Purkinje cells based on the error information that they receive. And imagine, this is just pure speculation, that anatomically, the ones that get information from the same error region should be placed together. And then if we did that, then their spikes would make sense, which so far they haven't done. Okay, so we're going to organize the Purkinje cells into populations. Step one, we measure how each Purkinje cell responds to a prediction error. Those are the complex spikes. So the monkey sits in a chair, looks at a fixation point. We present a target. After the target is reached, we jump the target to some location. Say it's jump to the right or jump to the left. So if it is jump to the left, here's the examples of it. You see these red guys here? Those are the complex spikes. You see that it's generating when the saccade ends at time 0. Afterwards, there's some group of complex spikes that are generated. When the jump is to the right, you see there's hardly any complex spikes that are generated. So this Purkinje cell cares about errors to the left, not so much errors to the right. You can measure its tuning to error like this. And what you see is that, yeah, it prefers errors to the right. Fine. So what we found is that for this Purkinje cell, it was getting error information from a particular part of the superior colliculus. It cared about, in this case, leftward visual errors. There it is. It generated complex spikes after this sensory prediction error occurred, and it cared for something that we're going to call CS on. CS on is the direction of error that engaged this Purkinje cell. If we do it in the opposite direction, you see it suppresses the complex spikes. So what we have done is that form a tuning based on this complex spike activity, and we've defined its preferred error. All right, now what we're going to do is do a coordinate system transformation. Use that error preference to define a new coordinate system for every Purkinje cell. So you measure activity of Purkinje cells in the cerebellum, and you see that some care about the ones on the left, some care up, some care right, and so in this case there's some sampling of Purkinje cell preference for error. Now what you do is that for every Purkinje cell, use its preferred error to define a new coordinate system. What do you mean by that? So suppose there's a Purkinje cell that cares about leftward errors. That's the direction of CSO. Now you present a target like this at 45 degrees. Well, why do you call this 45 degrees? Well, because we have this arbitrary thing that we say 0 is here, right? Suppose that we don't do that. Suppose we rotate our angle so that 0 refers to the preferred error of this cell. Okay, so now this is not a 45-degree saccade. This saccade is CS plus 250. CS is here, 250 is this. All we've done is done a rotation of the coordinate system based on each cell's preferred error. All right, so I'm going to show you this solves our problem. Okay, let me show you what's going on. So remember, we started with this problem. You have a saccade that looks like this, and then you measure cells. Some do this, some do this, some do this. So it's all over the place. They don't seem to make any sense. Now, what you do is that you form a population. You organize the cells into groups that share a similar preference for error. If you do that, what you see is that, well, there's some that burst, some that pause, but now you notice that when I say direction of movement, it's no longer in coordinate systems of the screen. It's in coordinate system of error. It's complex spike preferred direction, in this case, plus 180 degrees. Now, if I sum this two up, just simple linear summation of spikes, you see something remarkable. Look, you see a burst, then a pause, and then nothing. So these guys are canceling each other. You see this beautiful representation of the movement. So by doing a coordinate transformation based on error, clustering the neurons based on the error that they prefer, we get meaningful results. Now the spikes make sense. They are predicting the movement that's about to happen, a burst and then a pause. And I'm going to show you more about what this means. If you now look at it for other directions, CS plus or minus 90, CS on, you see that there's a difference between the activity of these cells. And the activity is encoding direction of the movement. We see also encoding of velocity and so forth. Now, let me focus on this concept of a burst and a pause. What's going on here? So Purkinje cells are inhibitory neurons. They represent the output of the middle layer. They're projecting onto the nucleus of the cerebellum. And what I'm going to show you is that this burst, which is increasing the inhibition, and then a pause, the pause is releasing the inhibition, is going to be beautifully timed to stop this movement, to decelerate the movement and stop the movement on time, providing us with a way to understand that the Purkinje cells are predicting when this movement should be stopped. So this
Can I have
encoding
one
of,
quick question?
yeah, of course.
So far you've been talking about directionality. Is there a tuning also in terms of the amplitude of the error along that direction? Is it just a
So,
spike
you know,
rate?
Yeah,
or-
yeah. So the error information was in the complex spikes, and all I've shown you was direction, right? We know less about amplitude coding. It appears to be much to do with the time of when the complex spike occurs. So the complex spikes that occur at about 100 milliseconds are really well timed to cause plasticity. The ones that occur a little bit later are not as effective as cause plasticity, and it seems like the probability of when they occur might be encoding the amplitude of that error.
Okay, cool.
Yeah, thank you for that great question. All right, so let me show you what goes on with these with the Purkinje cell population activity as a function of velocity of the movement. So these are saccades that are being made in direction CS plus 180, slow saccades, a little bit faster saccades, a little bit faster saccades, and what you're seeing is that this burst is growing as the velocity increasing, but notice that the pause that's occurring is right at the time of the deceleration onset. So the burst grows with the velocity of the forthcoming saccade, but the pause is time locked of when the movement is supposed to begin its deceleration and to be stopped. So it appears that the Purkinje cells are predicting when the movement should be stopped. So that's really critical, because remember, in that patient, what we saw was that the movements are overshooting. They're not stopping on time. This is making now some sense of what's going on. Let me summarize so far. We have trouble understanding activity of individual cells. In the cerebellum, what we did is that we said the Purkinje cells were organized into clusters, groups, based on the error that they received. When we organized the cells in this way, we found that the simple spikes made sense. They predicted basically the ongoing saccade. Now, the problem is that these Purkinje cells are inhibitory neurons, right? So they're inhibiting this nucleus. And it can affect the timing of when the nucleus cell fires through inhibition. But I'm going to show you there's something even more interesting. There's also synchronization of the spikes amongst themselves. So they can convey their information to the nucleus in two ways. They can change their firing rates, as I've shown you. Well, what they can do is that they can burst and then pause. And of course, by pausing, what they're doing is that they're releasing the inhibition so they can get the nucleus cells to fire. But they can do something even more amazing. These Purkinje cells, what they can do is that they can synchronize their spikes. So they are reducing their firing rates, but if they could synchronize the few spikes that remain here, they can make this nucleus cell fire even more. So how can they do that? Here's the result. So suppose that you have a group of Purkinje cells, inhibitory neurons, that are projecting onto this nucleus neuron. Avi Persson and Indira Raman, what they did is that in SLICE, what they showed is that if you just activate these neurons at some firing rate, say 50 hertz, but the cells are just firing randomly at 50 hertz, then what you do is that you just here's a nucleus neuron. It's just inhibited, just fires one or two spikes. So you just inhibit the output. So it's not very good. If all you want to do is inhibit, then your firing rate by itself can do that. But if you want the nucleus neuron to fire, then what you can do is that make your spikes synchronous. So if you have 50% synchrony of your Purkinje cells and all those spikes arrive, right afterwards, the nucleus neuron fires. Right afterwards, the nucleus neuron fires. So what you can do is that if you synchronize the activity of the Purkinje cells, you can make the nucleus neurons fire. So maybe the idea is that you not only want to disinhibit the nucleus neurons, release the inhibition, but whatever few spikes you got left, You want to synchronize it among the cells that you have. Okay, so this brings us now to the notion of why the heck do we use marmosets? Well, marmosets are small. They are primates, but they're small primates. What that means is that we can reach their cerebellum and use probes that were designed, silicon probes that were designed to be used in rodents, but because the marmosets aren't that much bigger than, they're basically the same size as same weight as a rat, you can use these specialized probes in marmosets. And what that means is you can record simultaneously from multiple Purkinje cells. That's the key. And so what do we see? What we see is that as this activity falls below baseline, as the disinhibition takes place, there is greater probability of synchrony. So remarkably, firing rate increases, but there's no change in probability of synchrony. However, during this special period of time when you're supposed to decelerate and stop the eye, now you see increased synchrony. And this amount of synchrony depends on the direction of the movement. So within groups that prefer a specific error, P cells combine their firing rates with synchrony to predict when the movement should be stopped. This is really a major result for us, because what it says is that the mechanism of computation, the way information is transmitted from the middle layer to the output layer, is not just the firing rates, but also the timing of when the cells are firing, and the cells are using temporal alignment of the spikes to communicate of when to stop this movement. So if that's true, then learning is going to involve not just changes in firing rates, but also changes in timing of when the spikes happen. A new direction for us to go. All right, but what does this have to do with learning and unlearning, Reza? I mean, you started this talk by talking about the honeybee and spontaneous recovery of memory. Now we've gone to the cerebellum, but we really haven't talked at all about learning it. So all you've done is to show me something about, how Purkinje cells, how we can understand the spiking of the Purkinje cells. All right, what does this have to do with learning? Okay, so what I'm going to show you is that the key is that specialization for error. That when you present an error in one direction, you have one group of Purkinje cells that cares about that error and will learn from it. But when you do extinction, what you're going to probably be doing is to involve a completely different group of Purkinje cells that care about that error. And these two are going to have interactions between each other, which potentially, I would speculate, might result in that concept of spontaneous recovery. Okay, let me show you what I'm talking about. So the idea is a single error will engage at least two different groups of Purkinje cells. So suppose I ask you to make a saccade like this, and I give you this error, right? Okay, so here's the cerebellum. Here's the area of the cerebellum that cares about visual motor control. And there'll be some cells on the left side of the cerebellum that care about those rightward errors. For them, that's CS on. And then what they're going to do is that they're going to learn from this error, and they're going to punish the Purkinje cells for the error, so the activity is going to fall. Early in adapt, it's going to be up here, down. Then later in the adaptation, their activity is going to be less. So this error took place. I care about that error. That's going to cause depression in the firing rate of those cells. But that same error to the right is also going to influence cells on the other side. So these guys care about leftward errors. So for them, that's going to cause suppression of the complex spikes below baseline. For these guys, it's going to cause increase in the complex spikes above baseline. For these guys, it's going to cause suppression below baseline. For them, there's going to be an increase in activity. So this is anti-preferred for them. It's preferred for these guys. So the same error causes activity in two different groups. Now, what's the difference between these two groups? I'm going to show you that the group that cares about that error, that prefers it, learns quickly. The one that is the anti-error learns slowly. I don't know if they forget at different rates, but they definitely learn at different rates. So the idea is there's fast learning in the cells for which that error was in the preferred direction, but there's slow learning in the cells for which the error was in the opposite direction, CS+1A. Okay, so this comes back to now the question of why is unlearning difficult? So we had this basic problem. We wanted to learn something, and then we tried to do extinction. And then what we saw was that there was a spontaneous recovery. What we did is that we looked in the cerebellum and we found that neurons could be organized into this concept of preference for error. Some cells care about leftward error, some care about rightward errors. And the reason why they care about errors that way is because the sensory space is mapped onto the colliculus, and there are regions where it cares about things to the left and it cares to the things to the right. So in that sense, it is really very different than a neural network where the error goes to everywhere. This is not the case in the cerebellum. Cells care about particular parts of the error space. So there is clustering of the error space. Each of these clusters, each of these Purkinje cells specializes in a specific error direction. A single error produces changes in many P cells, some preferring that error, others not. So for some, this causes an increase in the error complex spikes. For others, it causes a suppression of the complex spikes. Those that prefer that error cause an increase, learn fast. And those that don't prefer it, the ones that have preference for the opposite, they will learn slowly. When you reverse the error, look what happens when you reverse the error. So you had errors that were upward. That was the CS on for some Purkinje cells. Now you reverse it. You make this error here. What you're doing is that you are causing addition of basically this new group of Purkinje cells that prefers this error. So when you reverse the error, you are producing fast learning in a new group of Purkinje cells rather than strictly cause unlearning in the ones that were originally learned. So the differing rates of learning and decay in the various Purkinje cells may underlie this speculation associated with spontaneous recovery, something that needs to be worked out. Okay, so what's wrong with the work that I've been showing you? First, very important about the complex spikes, the center of this whole debate, this idea that I've been showing you. There's debate in our field about whether error is really encoded by the complex spikes. So we really need to do experiments that dissociate sensory prediction errors from other variables that may be encoded in this complex spike concept. The second thing that's a big hole in this presentation that I've presented to you is that I've been talking about these Purkinje cells. Well, the cerebellum makes its predictions not via Purkinje cells, but via these output neurons in the nucleus. I haven't talked to you at all about the nucleus neurons, what the heck they're doing. So we don't, just the same as we didn't used to understand the Purkinje cells, we know even less about the nucleus neurons. And so the reason why I didn't tell you about it is because honestly, we don't know. We don't know how to do populations. We don't know how to make sense of the neurons in the nucleus. So we need to do experiments, record from them, organize them into population, and maybe use this concept of preference for error to organize the nucleus neurons into populations. And finally, in the cerebellum, the predictions are conveyed via firing rates and synchronization. I showed you that the neurons do more than just change their firing rates. They also align the spikes in time. And this is really potentially the most interesting new direction to go, which is look at learning algorithms that not only change firing rates, but also change the timing of when a spike is going to occur. So organize the network in such a way that you make the neurons fire at a specific time. How do you do that? And if that's true, if synchronization depends on error, then we have a new learning rule, basically. That we have discovery of a new learning rule in the cerebellum, something that would be certainly quite interesting. Okay, let me summarize and finish. I've been claiming that it's hard to unlearn because many kinds of learning exhibit this concept of spontaneous recovery. Based on a simple theory that my students developed some 17 years ago, we can think about why there is spontaneous recovery based on the concept of fast and slow learning systems. Many systems exhibit this property. Maybe that's because of they involve multiple systems that are learning, some fast, some slow. What we did is that we figured out a task that showed spontaneous recovery, this saccade task. And then we found that there was a group of patients that had trouble with it. They couldn't learn. This was people with cerebellar deficits. Then we said, okay, what's the cerebellum doing in control of eye movements? What we had to do is to build a lab that measured these things in animals. So we built a marmoset lab. And my students and I decided on the marmosets because we thought they're really the future of neuroscience in terms of primate research, because they're small, because of their potential for use of these high density probes because of the genetic manipulations that may be possible in them. So we analyzed the activity of the cerebellum based on this concept of a population coding. The idea being that to make sense of the Purkinje cells, what we had to do is to organize them into groups that preferred a specific error. And so unlike a neural network where error goes everywhere, what we imagined is that there were clusters of Purkinje cells that preferred a particular error. And by understanding the error tuning of the Purkinje cells, we were then able to do a coordinate transformation to make sense of their predictions, their simple spikes. And when we did that, we saw that during decelerations, the simple spikes paused, went below baseline, but then had the synchronous activity between them. When an error occurred, one population of neurons learned slowly while another learned fast. And we think that this implication is that unlearning is difficult because a change in error engages a different group of neurons than those that initially learned, producing masking, but not unlearning. Okay, so I'm going to finish and I'm going to then turn the table over to you to ask questions. But before I do that, let me end with a poem by Rumi. It's just one line. He says, "Every storm the beloved unfurls, permits the sea to scatter pearls." Thank you so much for your time.
All right, thank you, Reza. It was a really informative talk. I feel like you explained things very clearly, so I really appreciate that. So I guess I'll end the recording here and we'll continue on for discussion as people see fit. Thanks, everyone.